In this video, we will continue with flux balance analysis and we will discuss the choice of objective function in detail and also understand the very interesting concepts of how there are alternate optima that are possible in flux balance analysis or in any optimization problem of this sort. So, now I want to draw your attention back to the fact that the solution is unique or the objective function value is unique, whereas the possible flux distributions are many. So, let us go back to the math of it. Where did we start in the previous module? Let us say this is V1, this is V2, this is V3. We put in some linear constraints here, and these constraints will basically give you what is known as a flux cone, right. These are all your linear constraints, right, and these are your possible solutions, and if you have more constraints, maybe one of these is the optimal point and so on, right. But for each of these optima you can, all of these may be equally good, all of these may be equally good optima, right. You can obviously say they have different values for V1, V2, V3, but they might end up having the same objective function. In fact, in linear programming they will end up having the same objective function. If you have one best value, that is what you will always get. So, what does this mean if we go back here? So, here you clearly got it, I will repeat it one last time. You can have 0 0 0 0 10 5 5 10 10 10 as one possible solution, or you could even have 10 10 10 10 10 0 0 as one another possible solution. These are two extreme solutions and a bunch of solutions in between, right. But beyond that, what does it mean when we look at the objective function? Let us say you are representing a likely physiological objective or you are representing a bioengineering design objective. These are two classic cases where you will use FBA. Why would you use FBA? You have a cell and you want to predict what is its growth rate in a particular medium. You can use FBA, right. You compute max C transpose V, that is basically some V biomass such that you get the max possible growth rate, and under ample nutrient conditions usually the cells grow at their maximum growth rate. So, this you find that it usually agrees very well in practice. The other would be max production of, let us say, lycopene, which is some secondary metabolite and important nutraceutical and so on. You will invariably see that, so as somebody once told, S to X is the life of the cell, right. Substrate to biomass, S to P is life of metabolic engineer, right. The cell wants to do S to X, but you want to somehow pull it and do S to P. You do not want to totally kill this off too, the cell is dead, you cannot have any product. So, you want to strike an optimum somewhere here, right. But now if you say maximize production of lycopene, what is the use of the flux distribution that you get here versus the use of the flux distribution that you get here? First thing, this flux distribution, if you multiply it with your objective function, you will get the growth rate, the correct growth rate, but you already have been convinced that you can have or let us use a you can have many equivalent flux distributions. They are all equivalent, they are not equal, but they are all equivalent. They will all give you the same biomass. This means that the same reaction can vary in flux, can occasionally admit more flux or less flux, but still have the same biomass. So, they can compensate. A very simple example would be, let us say you have A, X, this is one pathway, this is one pathway. So, all the flux can go like this, all the flux can go like this, or some fraction can go in either way and you can still get the same output, right. So, you can have different values for the flux distribution, different flux distributions that compute the same growth rate in practice. Does that make sense? So, let us say and let us say your objective function is maximize V1 plus V2 plus V3 and this was a solution that you got, right. So, your function value is basically 1.4. There are obviously other solutions that are possible. You can have 0.4, 0.4, 0.6, 1.0, 0.2, 0.2. All these will basically compute the same optima, which is 1.4. So, this may mean that more flux is going through this branch, less flux is going through this branch, whereas this might mean that all the flux is going through this branch and very little is going through this branch and so on, right. So, in different scenarios this is going to be the same in an LP, whereas this can vary. So, in other words, you are very confident of the FBA prediction, the growth rate prediction from FBA, whereas you are not that sure of the flux distribution that you obtain from FBA. It is only one of the many possible flux distributions. So, there are ways to, so one way you can say is if you want to further constrain, make it a unique distribution which is more likely, you can say can you find me minimize the sum of all fluxes. In fact, if we did this on this problem, we would basically get 0 0 0 0 10 10 10.
Then C or
No. So, this is an additional constraint. This does not replace maximize C transpose X, maximize C transpose V. So, you say maximize C transpose V and of now when you do this you might get many V's, but the same value for C transpose V, same value of objective function. Is that clear?
But C has only one non-zero term.
See, that is in this example, that is not usually the case. So, but even in this case, so that is very good, right. So, if C has only one non-zero term,
Then there's only one answer.
so, no, the zeros can multiply anything, right.
No, there's only one answer for that flux in the vector.
Yes, okay, fair enough, yeah. But it's very easy to imagine that C into V1 is also equal to some, let us say 0.8 is your growth rate, C into V2 is also, and this V1, V2 and I need to use slightly different notation, right, V1, V2 are elements we will consider. So, 0.9 and C into V triple prime may also be 0.8, but that is so simple because there are so many C has so many zeros, they can multiply with any number, it is still not going to affect your final dot product, right. But anyway, that is digging into the nitty gritties. But the other thing is, on top of this is like the example I was telling you in the morning. Solve x plus y equals 10 such that you also minimize x minus y or something.
In this case, do you take the.
So then it will immediately give you, so x plus y equals 10, I will get many solutions, 10, 0, 3, 7, 5, 5, 7, 3, 10, 0, 10. Now if I want to minimize x minus y, it becomes this. So of all the flux distributions that you get me the one that has the least absolute sum of the fluxes or the least number of non-zero fluxes. We will come back to this at a later stage, it is a very interesting point. So this is in fact called L1 norm minimization. We will come back to this in much greater detail at a later point. So now to come back to this, a very interesting consequence of this is, what does it mean when I say to represent a bioengineering design objective? It is the same as saying maximize production of lycopene, right. So once again let us repeat this. What does this mean? These are possible states of the cell. This means that reaction 1 in the cell is going to go at the rate of V prime element 1, reaction 2 in the cell is going to go at the rate of V prime element 2 and so on, right. So this means that the final growth rate is going to be V bio, but it can be any of these possible configurations the cell can take. What does this mean on the other hand? I say maximize production of lycopene, it will tell you what is the theoretical maximum lycopene that is producible by the cell. What about the flux distribution? Can you attach any significance to it?
Now you want to change your cells.
Potentially, right. So you have, so currently the what FBA will give you is, it will give you one configuration of the cell which can give you the maximum lycopene production. You can now see how far that is from your current configuration. So you can say this is your current configuration and this is the alternate configuration, and how far are these two distributions, right. So potentially you want to reroute, you want to induce, provide suitable inducements to the cell in terms of overexpressions or knockouts to pull it closer to this reality, to this desired reality. So this is the desired reality, right. More of the substrate is going towards your product of interest than towards the cell's product of interest. So is that clear? So it is very important to understand that there are alternate optima, right. So in any modeling exercise, it is important to understand that, to interpret your, to be able to interpret your results. So with confidence you can say that my FBA solution is my expected growth rate of the cell under whatever nutrient conditions we have specified, right. Whereas the flux distribution is one of many possibilities. We do not know which one of them the cell might actually be inhabiting, so to speak. But when you do the same thing for say lycopene or some secondary metabolite, it turns out that the solution that you get may be one possibility that will give you maximal lycopene, and the one useful part is whatever max lycopene you get is the theoretical maximum. If you know that your cell, if your current biotechnological process is already producing 90% of that or 95% of that, you may not want to worry about it, right, because there might be some non-ideal idealities, and this is anyway, no model is perfect, right. But on the other hand, if your current biochemical bioprocess is producing at say 60% of the theoretical maximum, it is a very fertile ground for further investigation to see what kind of manipulations you can employ to get the cell to produce more of lycopene or whatever product of interest. You should be able to quantify that. Maybe you are putting in a couple of gene deletions, couple of overexpressions, I can simulate that. We will see how to do that in like the immediate next class. In this video, we had an overview of how we go about picking the correct objective function, be it for overproducing a metabolite or maximizing the growth and so on. And we also looked at the concept of alternate optima in flux balance analysis. In the next video, we will see how we can sort of circumvent the need for maximization of growth rate and use a slightly different objective function which turns out to be quadratic. So, this technique which we will discuss about next class is known as minimization of metabolic adjustment.